Hey everyone, thanks so much for joining us today for the Single Cell Spatial Analysis Program seminar. Happy Juneteenth. So I'm Justin Colcino, I'm one of the PIs of SCSAP, and I'm really honored to be here today with Ying Ma. For those of you who may not be aware, SCSAP is a program trying to unite folks around this idea of single cell and spatial omics analyses. If you are not yet a member and you would like to be, if you're in person, touch base with me or Evan or Vanessa after the seminar, or Vanessa, could you drop a link to the our website in the chat, please? And so today we are lucky to be joined by Ying Ma. So Ying just defended her PhD in biostats here at the University of Michigan School of Public Health last month. Congratulations. And Ying has been doing like incredible work in the area of statistical genetics, but more importantly for our seminar series, just like exceptional work in this area of spatial omics data analysis, this like really hot area. And I think Ying is now nationally recognized as she is going to be starting a faculty position at Brown University in their Department of Biostatistics. Very excited for this. And so I'm going to turn it over to Ying to talk about cell type deconvolution in spatial omics analyses.
Thanks for Dr. Colcino's nice introduction. And today, I'm very honored to be here to present my recent work on spatially informed cell type deconvolution for spatial transcriptomics. So here is the outline for today's talk. I will give an overview of the spatial transcriptomics. Then I will talk about the cell type deconvolution in spatial transcriptomics, and also including our developed method card, and also its demonstration in both simulations and real data applications as an example. Finally, I will give you guys a summary. So as we know that cells are the basic unit of life, for example, here is the brain tissue. Such biological tissue is a group of cells that have similar function and structure together as a unit. So the distinct cell types differ in their both structure and function, which is usually related to their gene expression. And different cells may express very different sets of genes at different levels. And the set of genes expressed in a cell often determines their unique properties, as it is shown here. And understanding the cellular heterogeneity characterizing the tissue organization may provide valuable insights into the biological mechanisms, as well as for the therapeutic targeting for distinct diseases. So how can we measure the gene expression? Here are three technologies that we can, measure the gene expression. So the first one is the bulk RNAseq, and second, more recently popular single-cell RNA sequencing and spatial transcriptomics. So next, I will briefly introduce what they are. There are multiple ways to measure the gene expression. The traditional way to measure the gene expression is the bulk RNAseq, which actually measures the average gene expression across a bunch of cells, which might belong to homogeneous or heterogeneous cell types in this sample. And however, this might hinder the single cell resolution. And this is the analogy of this technology for us to better understand this. So we can imagine that it is like a painting with a mixture of dots of colors, but we don't know what the exact color is for each dot. And the data format is usually the average expression count data across all the cell types in this sample. Then the single cell RNA sequencing is rapidly developed over the last 10 years. So different from the bulk RNA-seq, it is first to isolate cells and then enable us to measure the gene expression profile for each single cell. So we can use this color palette as the analogy to understand this technology. Now we know the exact color of each dot is, and each color tone can be corresponding to the cell types, and the dots of the similar colors can be categorized into the one cell type. And the data format is usually the count matrix, which measure the gene expression profile for each individual cell. And the single cell RNA sequencing data is usually sparser and noisier due to the technical factors. However, the single cell RNA sequencing cannot capture the spatial information of cells due to the isolation step. Thus, leads to the failure in the understanding of the organization of cells in the tissue landscape. Hence, the spatial transcriptomics has been developed over the last 5 years. It can be viewed as a pointillistic painting. So in the pointillistic painting, the dots of colors applied in patterns to form the imagery. So now we know the exact position of each dot in this painting. And the data usually consists of two data matrices. One is the gene expression count data, measures the profile for each spatial location. We call it spot here. And correspondingly, we also have the two-dimensional coordinates to measure the spatial location. And however, this for most sequencing-based technologies, for each spatial location, we actually don't know what are the cell type compositions, what are the percentage of each cell type in this spatial location. And the exact role of the tissue in an organism depends on what types of cells it might contain, especially for the tumor tissue. For example, here is the pancreatic ductal adenocarcinoma, and which is an extremely lethal disease with more than 45K estimated deaths in the USA, according to the statistics in 2019. And the microenvironment of this PDAC often consists of multiple cell types, for example, the fibroblast cells, immune cells, pancreatic stellate cells, and the extracellular matrix. And this complexity of the tumor microenvironment promotes the tumorigenesis, disease development, and metastasis. And understanding this tumor microenvironment is very important for the PDAC treatment. And estimation of the cell type composition is a crucial step towards the identification of the cellular target of the disease, as well as also the analysis of the tumor heterogeneity and the prediction of the response to the immunotherapy. So with the spatial transcriptomics data available, we can actually investigate how the cell type compositions vary spatially. So this middle figure is the results of CARD as a motivating example. We can see clearly that the cell types are distributed, clearly show the structure of this PDAC tissue. And we will talk more details about this example. And with the known cell type compositions for multiple tissue slices in the future, we can also compare how the cell types change between the individuals belonging to different treatment groups. So next, I will talk about our method CARD for spatially informed cell type deconvolution for spatial transcriptomics. So here is a simple logo to show what task that we want to perform. So basically, CARD can estimate the cell type composition for each spatial location measured on the entire tissue. So here is a recap of what spatial transcriptomics technology is. So briefly, spatial transcriptomic technology enables the gene expression profiling on a tissue sections with spatial localization information. And there are several spatial transcriptomics technologies available. One of the most popular platform is the 10x Visium. So here is the simple illustration of the 10x Visium. So first, you will get a commercially available slide with about 6 different squares, and each square containing about 5,000 barcoded mRNA props. And then you overlay a specific tissue on the square, and the mRNAs released from the tissue will be hybridized with the barcoded props underneath. So these barcoded props enables you to measure the gene expression for each spatial location. And at the same time, there are also spatial barcodes, which contains also the spatial information. So at the same time, this will also allow you to obtain the spatial information. So again, through these spatial transcriptomic technologies, you will get a set of 2 data matrices. Specifically, one is the N by P count matrix of the gene expression, where N is the total number of spatial locations on this tissue, and P is the number of genes. And another one is the 2-dimensional spatial location matrix with these 2-dimensional coordinates. And however, what I introduced at the beginning, there are several challenges in spatial transcriptomics data analysis due to the technical limitations. So the first challenge that I've mentioned is that because the transcripts captured at each spot contains a mixture of cells for most of the sequencing-based technologies, and the cell type composition in each measured spatial location is unknown, there are unmet needs to understand the heterogeneity of cell type compositions in the tissue samples, especially for the cancer tissue, as I introduced at the beginning. So the first contribution of CARD is that it can provide, estimate the cell type proportions accurately. So here, imagine that this is a specific spatial location. We can estimate the cell type proportions for each of the spatial location measured on the tissue. And the second challenging is that the technical challenges lead to the insufficient spatial resolution of the unmeasured spatial transcriptomics data. For example, in the 10x Visium data of the pipeline that I showed, each measured spot is actually at the spot level, which might contains about 10 to 20 cells. It is not at the single cell or the subcellular resolution. So it's imperative to reconstruct the high resolution map based on the low resolution transcriptomics. And all these challenges motivate me to develop the new method, CARD. So the second contribution of CARD is that it can reconstruct the high resolution map based on the low resolution data to solve the technical limitations of most low resolution sequencing based technologies. Actually, the spatial transcriptomics technologies faces a dilemma of knowing the location of transcripts, but it does not know what cell that produced them. Well, the single cell R sequencing data enables us to measure the gene expression on hundreds of thousands of cells from different cell types, and thus can provide these valuable cell type specific information. So given this set of complementary strengths and weaknesses, the idea of combining these two data together to infer the cell type composition at each spatial location is very compelling. Indeed, the single cell R sequencing data have been previously used as a reference for deconvoluting the bulk RNA-seq data, because the bulk RNA-seq data is also a mixture of cells in each sample. And in principle, people might think that these methods can be directly applied for the deconvolution on the spatial transcriptomics data, just treating the data for each spatial location as a bulk sample. However, this direct application is not capable in spatial transcriptomics because, first, the number of cells within each spatial location is much smaller than that of the number of cells in each bulk sample. And second, the spatial transcriptomics data often contains more spatial locations than the number of samples included in the bulk RNA sequencing data. So such direct application is not optimal. And since 2020, a few deconvolution methods have been specifically developed for the spatial transcriptomics data deconvolution. For example, here, the RCTD, which was published on Nature Biotech 2021, it utilizes the Poisson factor analysis model to model the expression count from spatial transcriptomics data directly with the mean cell type specific gene profile estimated from the single cell RNA sequencing data. And however, all these current methods, they have some limitations. First, all these methods actually ignore the spatial localization information, which might be useful or also reflect the biological fact. And second, all these listed deconvolution methods, they cannot reconstruct the high resolution map. They can only deconvolute the cell types and generate the estimated proportion on their original resolution. And the third, all of these methods, they only implemented in their method, which requires a single cell RNA sequencing reference data, which might lose the robustness or also be sensitive to the choice of the reference data. So here, in particular, we know that a tissue is often composed of multiple cell types that are segregated in a spatially correlated fashion into tissue domains during their development. So these tissue domains are actually characterized by their region or domain-specific composition of cell types, with similar cell types are colocalized. So consequently, neighboring locations on the tissue likely contain more similar cell types than the locations that are distant, leading to the correlation in the cell type compositions might in the distance-dependent fashion. So this first figure actually displays the median proportion of the ependymal cells within the n nearest neighbors in the Murphy's dataset, which is a single cell resolution dataset. So it actually indicates that the similar cell types tend to colocalize with the colocalization pattern decaying with the distance. And also the analogy here we can see from this pointillist painting is that we can see that similar color dots locate nearly that they usually form a specific structure like the flowers and bushes and finally form the image. So that's kind of analogy for you to understand the importance of the cell type, the similarity structure in the cell type composition matrix. So here we introduce our method called CARD, which is short for conditional autoregressive based deconvolution. Briefly, CARD is the main version of CARD is a reference-based deconvolution that actually leverages the cell type specific gene expression information from single cell RNA sequencing data for the deconvolution of the spatial transcriptomics data. And a key feature of CARD is its ability to accommodate the spatial correlation structure in the cell type composition across the tissue locations. So the result is actually the estimated cell type proportion matrix, with each row represents the proportion vector for each spatial location or the spot here. And after we estimated the proportion, we can jointly visualize this using this scatter pie plot, and we can clearly depict the structure of the tissue. And each small pie represents a specific vector of the cell type proportions. And a unique property of CARD is that it modeling the spatial correlation structure. So utilize this characteristic, we can actually impute the cell type compositions on the unmeasured locations, utilize the spatial correlation relationship between the unmeasured locations and the measured locations to reconstruct the high resolution map to potentially provide novel biological insight that was not provided by the original resolution data. So let's briefly look at the model details. So specifically, CARD builds upon a non-negative matrix factorization model to link the cell type specific expression profile B matrix, we call it reference basis matrix, with the spatial transcriptomics data X. So the detailed biological interpretation of this basic model can be seen in previous deconvolution papers. And specifically, X here is the G by N gene expression matrix measured on the N spatial locations. And B here is the G by K reference basis matrix measures the mean level of the cell type specific gene expression. And V here is the V by K cell type composition matrix that we want to estimate. And E here is the G by N residual error matrix. So to accommodate the spatial correlation structure, as I mentioned in the motivating example, you're showing the decaying pattern in the cell type composition matrix V here, we specify a conditional autoregressive modeling assumption on each cell type K. So actually the CAR modeling assumption effectively expresses that for each spatial location I, its case cell type is a weighted summation of the case cell type of all the other locations. And here the BK represents the case cell type specific intercept that represents the mean level of the cell type. And W here is the weight matrix between the spatial locations, and we constructed using the two-dimensional coordinates. And phi here is the spatial autocorrelation parameter that determines the strength of the spatial correlation structure in the cell type composition. And epsilon IK is just the residual arrow. So this CAR modeling assumption equivalently to say that conditional on the cell type composition for all the other locations, the proportion for ith location is following a normal distribution with their mean is a weighted summation of the proportion on all the other locations. And we use some mathematical derivations. We found that the joint distribution follows a multivariate normal distribution with specifically the variance covariance can be expressed as a function of the weight matrix and also the spatial correlation strength parameter. And specifically, we follow previous literature to construct the weight matrix W. And here the using the Gaussian kernel, and SI and SJ are the two-dimensional coordinates. And we fix the scale parameter for simplicity and also for computational simplicity and also for the sensitivity analysis we observed. And then I developed the constraint optimization algorithm in the maximum likelihood framework to estimate the cell type proportion V. Specifically, I performed the optimization based on this log likelihood. And I will not go details about this, but I want to give you an overview of this. What are the objective functions we want to maximize? So this actually likelihood actually consists of three parts. So the first part is the reconstruction error linked between the spatial transcriptomics data and the single cell reference data. And the second part just models the spatial correlation structure, assuming the CAR modeling assumption. we assume that neighboring locations contain more similar cell type compositions. And the third part is our assumption about these hyperparameters such as the cell type specific parameter. And the CARD's main modeling assumption actually relies on CARD main model version relies on the reference data, and where the reference B matrix basis data B is constructed from the single cell RNA sequencing data. And this can be following the MUSIC's framework, which is a bulk RNA-seq deconvolution paper. However, people might question sometimes when the reference is not available or the single cell reference data quality is not that high, how can we use your method CARD? So we also extend CARD into a reference-free version. So in that reference-free version, we call it CARD-free. In that version, we don't need to rely on the single cell RNA sequencing data. Instead, we require the user to input the list of the marker genes, and we will iteratively estimate and update the reference basis matrix based on the marker gene. So I have performed extensive simulations and also real data applications. Before we look at the simulation results, let's first check whether our simulated data reflects the characteristics of the real data. So the left panel shows that the simulated data are realistic, preserving data features observed in the published spatial transcriptomics data. And the two different shapes represent, one is simulated, one is a real mouse olfactory bulb data. We can see that it preserved this, the data characteristic. And the middle panel displays the simulated proportion of one setting as an example. And we see that it actually mimics the layered structure of the real mouse olfactory bulb tissue. So we think that it's really important for us to first check our simulated data, whether to ensure our simulations reliabilities. And then we perform the deconvolution using the simulated data, and we quantify the deconvolution performance by computing the root mean square error between the estimated cell type composition by different methods, as well as the true cell type compositions in the simulation. And we further contrasted the RMSE of other methods with respect to that of CARD by computing these RMSE differences to remove the unnecessary difficulty level variation across all the replicates. So an RMSE on the y-axis below 2.0 indicates that CARD's RMSE is smaller than the other methods, which indicates a higher deconvolution accuracy. So here we perform one baseline setting and 4 challenging settings. So for the baseline settings, we split the single cell RNA sequencing data into 2 halves. Use the first half to simulate the spatial transcriptomics data, and use the second half as a reference to deconvolute for each method. And we found that CARD can outperform of all these state-of-art methods. Also, we also evaluate the following challenging settings because we want to see whether the different methods are sensible to the choices of single cell reference data. So we miss one cell type at a time in the reference data to see whether the method is sensible to the missing information in the reference data. We also add one more cell type to see whether the method is sensitive to the redundant information in the reference data. We also artificially create a misclassified labels in the reference data to see whether it is sensible. And finally, we also evaluate if we use an external single cell reference data, which generate from different platform, but contains the similar set of cell types, whether they are still sensitive and whether CARD can perform better than other methods. So clearly we see that CARD still perform than other methods. And it seems that in the challenging settings, the performance gain is much obvious than the baseline setting. Besides these, the more setting, which might indicate that these deconvolution methods are not sensitive to the redundant information, but more sensitive to the missing, misclassified, and the similar reference from an external study information. So next, I will use the real data from the human pancreatic ductal adenocarcinoma to showcase the accuracy and the utility of CARD in this cancer tissue. So the PDAC is the most prevalent neoplastic disease of the pancreas, and its complex tumor microenvironment and the intratumoral heterogeneity make it very challenging to review the underlying mechanisms. here is this HE staining image that was annotated by the pathologist from the original publication. So we can see that it mainly consists of 4 regions, including the cancer region in the right part and the ductal epithelium region colored in orange, and the pancreatic region colored in blue, and also the interregion is the stroma region. So I compare our methods with current state-of-art deconvolution methods. So here are the results of joint visualization of the cell type compositions. Specifically, we found that the cell type compositions estimated from CART can accurately review the heterogeneity of the PDAC tissue as it was shown in the HE image. And CART shows a clearer structure of this tissue than the other methods, which matches with the pathologist's annotation on the H and E staining image. So for example, we found that 2 cancer clone A cells and cancer clone B cells colored by the pink and green are highly enriched in the specific cancer region. And more importantly, CART is the only method that can further divide the right cancer region into 2 subregions that are missed by all the other methods. So we found that the upper region is more dominated by the cancer clone A cells colored by the green and colocalized with the cancer clone B cells colored by pink, while the bottom region is more dominated by the cancer clone B cells colored by pink. So these results actually, thank you for your guys' patience here. So next, I also checked the spatial distribution of each cell type separately and whether they matched with their associated marker gene patterns shown in the second panel. Consistent with previous scatter pie plot, CART can localize these cell types into specific tissue regions. So as is also evidenced by the marker gene patterns. And so the first plot actually displays the distribution of each cell types separately. And the second row displays the gene expression pattern for these marker genes. Importantly, the marker gene S100A4 is actually an important prognostic marker for the early stage pancreatic cancer. And its expression is found in previously studies to be associated with poor clinical outcome of patients with the pancreatic cancer. And I found that the S100A4 positive cancer clone B cells is highly enriched in the bottom region, which is different from the cancer clone A cells, which is highly enriched in the upper region, might suggesting that the bottom cancer region might be likely an early cancer region. And then I found that our findings are consistent with the previous pathologist annotation and can also provide novel biological insights. Specifically, we compare the differences of each cell type distribution between the pathologist annotated cancer and non-cancer region. So besides the high enrichment of cancer cells in the pathologist annotated cancer regions, CART is the only method that can review the heterogeneity of the distribution of these 2 microphage subpopulations in the cancer and non-cancer regions. So specifically, we found that the microphage B is highly enriched in the non-cancer region, likely indicate its function as a tissue resident microphage, while the microphage A is not statistically significant differently distributed between these 2 regions, likely indicate its high inflammatory status. So this heterogeneity, the distinct distribution of these 2 subpopulations of this macrophage actually represents a key functional cancer compartmentalization. And importantly, although we don't know the truth in this real data, but we can quantify it indirectly. So ideally, we expect that the mean cell type composition for these spatial data should be highly correlated with that measured in this sample matched single cell RNA sequencing data because they are from the same patient. And we compare this correlation, we found that CART substantially outperforms than the other methods, generates a higher correlation, again, supporting the accurate deconvolution of CART. And besides revealing the cellular heterogeneity of the tissue, CART is also able to reconstruct the high resolution map based on the low resolution spatial transcriptomics data. So this panel shows the high resolution map of the cell type compositions for the selected cell types and found that CART review a refined boundaries between different tissue subregions. For example, in this high resolution map, we found that the cancer clone B cells is more obviously enriched in the subregion, which matches with our expectation. And consistently, we can also see that the imputed gene expression levels by CART can also reveal a stronger spatial pattern than the original expression data might due to the technical issues that may not show the obvious pattern. For example, we can see the S100A4 gene. It's in the high resolution data. It has a more stronger enrichment in this subregion. So finally, then we also apply our method to analyze a high resolution spatial transcriptomics data, because in the PDAC data, it's still a low resolution data. And in this high resolution Slide-seq version 2 data, it is more challenging and it contains more spatial locations, which is about 53K locations, and can cause a huge computational challenging. And this high resolution data usually for each location only contains about 1 to 3 cells. And the data set is very sparse and contains more locations than the previous ones. And we found that CART correctly identifies the hippocampus region, since this is the high resolution data, and each spatial location is usually containing about 1 to 3 cells. So we can actually quantify the deconvolution performance by examining the expression levels of the marker genes on each of the 3 hippocampus regions. So we reason that a good deconvolution method will yield accurate cell type composition estimates and subsequently accurate tissue structures, and would thus capture the expected structure specific expression pattern for these marker genes. For example, the WSFS1 gene is a marker gene for the CA1 cells. It should be highly enriched or expressed in the CA1 region. And then we compared CART with different methods on their inferred CA1 region. And we found that CART generates the highest expression of this marker gene. Similar consistent conclusion we can draw from these CA3 cells and the dentate gyrus cells. And we found that CART can accurately infer the CA1, CA3, and dentate gyrus cells regions more accurately than the other methods. And as I mentioned at the beginning, most of the current deconvolution methods, they mainly rely on the single cell reference. So we also, to release such assumption or to release such choices of the single cell RNA sequencing reference, we also extend CART into a reference-free version. So in the reference-free version, we only require users to provide a list of the marker genes. So this is the results displays the result, the reference-free version. And we found that generally it can display a comparable pattern with the reference-based version, even if it utilizes less information. And there are 2 advantages of the reference-free framework that I'd like to mention. First, it does not require the single-cell reference data, so it does not limit it to the choice of the reference data. And second, it is not limited to the number of cell types existing or pre-annotated in the reference data. And also, we also extend CART into another single cell mapping. And this is actually still a remaining question. So the single cell mapping means that because at each spatial location, it is at the spot level, it's not at the single cell level. So people want to extract what are the specific expression profile for each single cell distributed in this spatial location. So we also extend that. So this is the result of the estimated single cell profile. So each small dot represents 1 single cell. But I'd also like to mention the limitations of this. So it's also a still remaining opening question, because it is very challenging to infer the exact position of the single cell. So in the future, if we can incorporate HE image information, maybe extract the single cell position information, and then combine that with this CART extension framework, that will be more interesting. But yeah. And finally, CART is computationally fast and memory efficient compared with all the other methods. So for the low resolution data, PDAC I introduced here, it only takes about 10 seconds and 3 megabytes. But for the high resolution Slide-seq version 2 data, it only takes about half an hour to finish the whole analysis. And yeah, to summarize, CART is a method for spatially informed cell type deconvolution for spatial transcriptomics, and there are 4 main contributions of our method. First, it provides accurate and robust deconvolution. Second, it can reconstruct the high-resolution map. And third, it is not limited to the choice of single-cell reference data. You can use our reference-free version. Fourth, it is really computationally efficient and user-friendly. And I also found that CART is listed as the top 3 methods in a recent Nature Communication benchmarking paper. And the software is available on my GitHub website, and the manuscript was published on Nature Biotech last year. And I'd like to give you an overview of the GitHub tutorial if you guys are interested to use that. So this is the CART tutorial, and there are the different sections about the introduction about CART and data input, and example analysis provides you the whole analysis of all the figures that you can reproduce for this PDAC data. And the experiments contains all the reproducible codes for all the other data applications and simulations. And installation just lists any issues you might need to pay attention to. So for the tutorial, it is very simple. You just require, if you want to use the reference-based version, you just require the spatial transcriptomics data and the single-cell reference data. And then just use some very simple line. For example, I didn't show here, for example, CART deconvolution, it will generate the proportion. And then I also provided a lot of visualization tutorials, also very simple line. You can use CART visualization pi, it will generate these, and a lot of options for the colors and also other arguments. So it's very flexible. And also I provide the code for visualize different cell types individually. So here just one line is CART visualize prop. And this is very interesting. That is actually motivated by a GitHub post. So I think a researcher wants to know, how can I visualize 2 cell types at the same time on the map? So I use this trick to first use the different colors and also use different shapes. So we can clearly see that the cancer clone B cells is located on the bottom region and cancer clone A cells is located on the upper region. And if we want to know what are the locations that have both high enrichment, we just need to find the location has a high red circle and the black square, so which can indicate there might be some interaction between these 2 cell types. And so I also briefly go over other methodology work. So this is the work which I designed to integrate the both DE analysis and gene site enrichment analysis together, because commonly these 2 analyses are separate. People first perform DE and then use the results from DE to perform the gene site enrichment analysis. So in this work, we call it IDEA. It can substantially improve the statistical power of detecting DE genes and gene sets, and also generated the calibrated p-values. And also, I'm currently focusing on another method called IRIS, which can help to detect the spatial domains for multiple tissue slices, and specifically focusing on large scale data sets such as Slide-seq and 10x Celium. And also, besides the statistical genomics work, I'm also working on some polygenic risk score analysis with Dr. Lars Fukistri and Bamar Mukherjee. So these are the work we published on American Journal of Human Genetics and Trends in Genetics. So we construct the polygenic risk score for commonly health-related exposures, and we prove that the combined polygenic risk score can help to predict the chronic conditions. So I will be joining as a tenure-track assistant professor at the Brown Department of Biostatistics and Center for Computational Molecular Biology. So feel free to contact me if you want to discuss with any research or you are interested in statistical genetics and genomics. And finally, I'd like to thank my advisor, Dr. Xiang Zhou, and all the Zhou Lab members. And thank you so much for listening, and thank you for your patience. Sorry for the technical issues, and any comments and suggestions are warmly welcomed.
Wonderful presentation.
Thank you.
This is very exciting. But one of the, I think, assumptions was that the similarity of cells together will help kind of guide some of the prediction of what the cell types are. We're showing similar colors together, right? What about for immune cells, which typically are isolated? would that mean that that card would be a little less effective in predicting that because those cells are isolated? So now that algorithm will be actually as active in predicting that aspect.
Yeah. Thank you for your question. So for the Zoom audiences, the question is because cards utilize the similarity assumption, but for the immune cells, typically they are isolated. So if when apply for the immune cell type deconvolution, whether the card is less effective. So my answer is because there was one key parameter is the spatial correlation strength parameter. We actually learned that parameter using a graded search from 0.01 to 0.99. So if it is a weak spatial correlation structure, that parameter will be very small. So that might ensure the effectiveness of our method. Yeah, thank you for your question. Yeah. Thank you so much, Meg. Yeah.
Have you seen if card is able to deal with other types of spatial omics data? Sort of like selfishly asked this, you just got a batch of spatial attacks. So have you like tried it in other besides like the Slide-seq paradigm?
Yeah, currently the question from Dr. Colosso is have I tried other types of the spatial omics like the spatial taxic or other omics data? So my answer is in my current the current application I haven't is mainly focusing on the spatial transcriptomics data. I haven't tried other technologies, but I think this modeling framework should be flexible to, for example, we can use some single cell based taxic data as a reference to see whether it is able to utilize to link that to the spatial ataxic data to see whether it can generate the biological meaningful results. But I think that's a really might be a meaningful application. But yeah, thank you for your question. Yeah. Yeah, Dr. Lee's question is the for the spatial weights matrix for the specific parameter that controls the spatial correlation strength, is that learned for each cell type or is that learned for each gene? Yeah, my question is that that is not learned for each cell type specifically, but we learned that we use the unified spatial correlation parameter strengths for all the cell types. But we have for each cell type structure, we also have the cell type specific scalar to differentiate different cell types. Yeah.
Well, great.